Yes.
Okay. And we tried dealing with the first and second, right?
Yes.
And we said that for the first question asking about explain why the students included the following. Okay, in the apparatus, the beaker of water and the thermometer. We try to give reasons for that. That beaker of water is to regulate the temperature, okay, to maintain the temperature. All right. Yes, that's one. And that the thermometer is to measure, okay, the temperature of the water, okay, to keep the temperature controlled or measured, right?
Yes.
Okay. We also answered this one that talks about the light meter and the roller. That why do the students use the light meter and the roller? And we have that light meter is to measure or check the light intensity, right?
Yes.
And the ruler, what is the one for ruler?
To measure the distance between the
Okay, yes, yes.
plant and
Accept ruler to measure the distance between the lamp and the plant. Okay, yes, to measure the distance between the plant and the light. All right. Yeah, I think that was how much we were able to do. Did you get to this part?
no, we didn't.
Okay, I think we are going to start from here. So I need you to supply the answers as appropriate for this. Let me make it a bit bigger. All right. Yes. So that's the first question. Describe the student's result, and the mark is 3 marks. Of course, you gain credit if you use data from the figure and the graph above. So let me hear your answer.
Um, the more carbon dioxide, what? The more carbon dioxide concentration there is, the higher the rate of photosynthesis, or the higher the rate of the bubbles per minute.
Okay, that's 1 point. Yes, that's 1 mark. There are 3 marks. Go on.
And as the line reaches to 0.40, it stays constant,
Mm-hmm.
meaning that the plant has reached its maximum amount of bubbles in um, chlorophyll.
Mm-hmm. Okay, at least you are quoting data now 0.40. Okay, and above, the graph stayed constant at region Y. Okay, that's 1 point. Yeah, one more. One more. What can you say at 0.0 concentration of carbon dioxide?
It was at its lowest. The rate of photosynthesis was at its lowest.
At lowest, even at 0.0, there was still some carbon dioxide available in the water, and so the rate of photosynthesis was lowest, and it was highest at 0.40. So you can equate the rate when it is lowest and when it is highest. Okay, so that's another value to say. So you can say from 0.0 all the way to 0.40, the rate of photosynthesis then stayed the same. It increased, and then so those are the things to note. If it's as much as 6 marks, this is just three. If they make it 6 marks, you have to keep pointing out every point, every details that the graph shows. That's how to describe. Describing does not mean explaining. Describing just means giving or telling what the graph, the result is displaying in words. Okay, in describing, you're only telling what the result is in words. That's description. So you have to take note of every details, every value, every data you quote, and you get your mark. So if it's 3 marks, you have to make sure you've highlighted at least 6 points. If it's 4 points, you highlight 6 points. When they say describe and explain, okay, that's when you start giving reasons for the shape of the graph. All right. So we know that carbon dioxide is necessary for photosynthesis. With the increasing concentration of carbon dioxide, the rate of photosynthesis is higher because more dioxide is available. Okay, to be turned into glucose and producing oxygen. Start giving reasons. That's the explanation. Okay, at 0.40, okay, at that point, carbon dioxide becomes a limiting, is no longer a what?
A limiting factor.
It's no longer a limiting factor. So you can now tell us at 0.40 that something else is limiting. You can mention that. So you can tell us that from between 0.0 concentration to 0.4, carbon dioxide is limiting. Okay, then above 0.4, it's no longer limiting. You tell us that the activity was highest at 4.0, it was lowest at 0.0. Those are the things to note when we are describing or explaining the graph. Let me go to the masking so you see what um, we have. Okay, so look at that now. It says rate of photosynthesis or rate of number of bubbles increases as carbon dioxide concentration increase. Then levels off. That's 1 point. Okay, it
Okay.
increases to 0.40. Okay,
Okay.
then above 0.40, it remains constant. All right, that's another point. What else did you see? They said it was slow at start. Okay, at the start it was slow up to 0.1. Then after 0.1, of course, it increased a bit faster. All right, so you can quote data. When you quote data of what you see, you get your points. So that's how to answer questions as this. Now look at this. They said state the factor that is limiting the rate of photosynthesis in region X. I think we've mentioned that already when we were explaining. What is the factor limiting the rate of photosynthesis at region X?
Carbon dioxide.
Carbon dioxide. Okay. Now, this one says suggest and explain the reason. Now, they are giving explanation. You are suggesting and you are giving the reason. Okay, explain the reason for the shape of the graph in region Y. Why is the graph taking a constant shape in region Y? Okay, with reason. You can do that now. That's 4 marks. So we need 4 points at least. Do I go to the graph?
Yes.
Okay.
Um. The the at line Y, um the rate of photosynthesis has basically reached its max in the plant, and what do I want to say?
Look at what they say. They say suggest and explain the reason for the shape of the graph in region Y.
Carbon dioxide is not a limiting factor anymore.
Yes. Okay. At Y, what is happening at Y? The graph is constant at what? Y. That's the reason. What's the reason now? CO2 is no longer a limiting what factor. And what is the reason? Why is that so? What is not limiting? So give reasons. What could be the limiting factors then?
Um, water availability, temperature. Um,
Go on. What are the first two things that should come to mind? When we talk about limiting factors, there are three things that are always the first things to mention.
temperature, light intensity, and
Thank
carbon
you.
dioxide
Thank you.
concentration.
So anytime they talk about in light limiting factors, please the first three to consider is light intensity, temperature, and CO2 concentration. So since in region Y, CO2 concentration is not limiting, what are the possible things that could be limiting? You cannot tell us that light intensity could be limiting. And what effect will that cause? It will reduce the rate at which photosynthesis can happen. It could be temperature. And how does temperature affect um, photosynthesis? Okay, it can affect the what? Enzymes, right? Temperature affects enzymes. Light intensity has to do with the amount of light the chlorophyll can what? Absorb. Do you get? So that should be your answer. So the suggestion is that at Y, the graph is constant at Y. Okay, showing that CO2 is no longer a limiting factor. All right. That means something else is limiting either light intensity as it's the amount of light the chlorophyll absorbs, okay, can affect the rate of photosynthesis. It could be temperature, because if the temperature is no longer suitable, enzymes catalyzing photosynthesis reactions will be what? Affected. So those are the things your answer should contain. And by the time you write those points, you will get up to 4 points for that. So you have to be objective in your answer. And anytime you are writing, you have to mind the space provided. It's not advisable to write beyond the space provided. So you have to really monitor, okay, or be aware and conscious of the space you have, such that you'll be able to list all your points, okay, to fit in within the space you have and get maximum marks. All these things are the things that you need to be mindful of to get your A star when you answer questions. Are you with me?
Yes.
So let's look at what they say. They say reference to limiting factor in a suitable context. That carbon dioxide is no longer limiting. Okay, that's one. Okay, reference to the fact that a factor is limiting and CO2 is not limiting. Then you can mention the limiting factor, for example, light or light intensity, and you tell us that light is needed to provide energy for photosynthesis. You can tell us that temperature is limiting and that temperature will affect the activity of enzymes that drive photosynthesis process. So these things are the things that actually would give you all the marks you desire. Each of those points, of course, takes a mark each. and you can see how the mark scheme is designed. So your answers must be objective. You don't need to make too much stories that are out of point. Okay, you must pick the point as concise as possible. Are you with me?
Yes.
Okay. So that's that with that. Okay, this question will also will also fly. Okay, let me hear you answer this one. Question C. Yes.
Um. Um. One one alternative way to measure gas is by using a gas um,
Remember, it's 3 marks, so you have to give 3 points around your explanation. That's what.
I'm trying to remember the name. Um, a gas syringe.
Yes, a gas syringe. So how do you apply it? That's just 1 mark. If you use a gas syringe, that's 1 mark. What are the procedures to adopt in using a gas syringe?
You you connect the syringe to um beaker or the object that you're containing the substance in, and
a lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have um, you know the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable, and you get the very best of service. We also have for the E levels as well, and also for the EdExcel. So we have for the Cambridge E levels and EdExcel E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for A level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com, and they'll reply to you. And the good news about this is, what I've discussed with the sales team is, we've tried as much as possible to see how we can subsidize the amount according to countries, so it doesn't really look very, very heavy. Okay, so so this is really important. Yes, we do charge in pounds, but again, we try to be as much as possible considerate to your countries, and we don't just skyrocket the price. No, because at the end of the day, we know how it feels, and we really want you to actually get the best of service, and again, be able to afford it from the comfort of your home. So again, Ajiro Tech private tuition is currently ongoing. You can enroll yourself for the IGCSE and then for the E levels. Again, for the IGCSEs, these are the subjects, and for the E levels, these are also the subjects. So again, what are you waiting for? Just send an email to the sales team. If you're an IGCSE student preparing for IGCSE, if you're watching this in 2025, when this is in 2026, when this is 2027, we're currently ongoing, and this is actually a good time for you to actually explore and then get it tailored to your own needs. So trust me, you don't want to miss out.
I'm giving you hints now from the things I'm writing.
You deliver the tube over water and use a
Okay, let's see what they said. You can measure the volume of gas collected, okay? Instead of counting the number of bubbles, you can collect the volume of gas or measure the volume of gas. How do you collect it? You can collect it using a measuring cylinder or a gas what syringe. And how do you do that? You collect the gas, okay, by displacement over water, all right? Or you attach the syringe to a delivery tube, okay, where you collect the gas. That's one way of doing that. So anytime you mention this point, you get 1 mark. This semicolon you see is telling you things that will get marks. You can measure or collect the volume of gas, 1 mark. How do you do that? You use an inverted tube or a gas syringe, another 1 mark. And you collect the gas over water, okay, or using a delivery tube to collect the gas. These are the points that are important. It's fine to know that you use a gas syringe, but you have to be able to give details of how it works. So you connect the setup to a gas syringe using a delivery tube or to a measuring cylinder to collect the volume of gas, measure it in a measuring cylinder, or you measure it collecting the volume using a gas syringe. If it's a syringe, you connect the gas syringe by upward displacement or downward displacement over water, okay, and you can measure the volume of gas collected. So these are the details to show that you know what we are talking about. Okay, do you understand?
yes.
All right, so I think that's the most we can take on photosynthesis, limiting factors, evidences to show that photosynthesis happen in plants and the likes. Okay. All right. Let me see. Okay, let's answer a few of these. Did you answer this question 26 last time?
No.
Can you give me the answer of 26, 27?
Mm,
I'm with you, Maribel. No, Mirabel, sorry.
um, uh, the cellulose.
Why go all the way to cellulose? But what did they ask? What does the leaf form first? What is
Oh,
first
form
produced?
first. Oh, so I read the question wrong. Starch.
Are you sure starch is what is formed first? Now, plants during photosynthesis, what is the first stable product of photosynthesis? Glucose. And that glucose, I told you, is transported as what? Sucrose. So plants make glucose during photosynthesis. Glucose is a sugar, is a monosaccharide sugar, all right? And they transport it as sucrose and they store it as starch. So with this explanation, what is the correct answer there?
Sugar.
Sugar. All right. This cellulose and starch, they are actually sugars, but they are complex sugars. And that's not the first thing that is produced during photosynthesis. So the sugar that is produced is glucose. This is only to confuse you, okay? Cellulose is what they use to make their cell wall. They convert the starch to cellulose, make their cell wall, or they can convert the sugars to protein. When nitrogen is added to it, okay, so when nitrogen is added to sugars, they make proteins from it. They can convert the sugars to cellulose and they use cellulose to make their cell walls. So the cell walls of plants is made of cellulose. Plants store their sugars as starch. Okay, they store their sugars as starch. They transport their sugars as sucrose through their phloem and they make their sugars as glucose. So the first product they make is glucose, which is a simple sugar. Then they convert the sugar to sucrose, a kind of disaccharide for transport. When they transport it as sucrose to the flame, they can then store it in other parts of the plant, either in the leaves or in other parts of the stem as starch for later use. Sometimes they can convert this glucose to other chemicals that they need, either cellulose to make their cell wall. They can convert them to proteins for their development, for cell repair and other things. They can convert it to starch too for storage. So that's what they use their glucose for. They can even use the glucose for
Creation.
respiration, okay, to release energy. So these are the uses of the food they make. They can ask you, what do plants use the sugar they make? What do they use it for? They can convert it to cellulose to make cell wall. They can add nitrogens to it to make amino acids or proteins. which is used for their growth or development. They can use it for respiration to release energy for metabolism. So these are the major things they use the sugars for. They can convert it to even sucrose for transport. They can convert it to starch for storage for later use. They can convert the sugars to cellulose to make cell walls. They can convert it to proteins when nitrogen is added to it. Okay, or amino acid which they use for growth and development of leaves, roots, and stems. You can use it for respiration to release energy for metabolic activities. Any question?
No.
All right, let's try question 27. I'm with you, yeah?
B.
Did you say B?
Yes.
Why did you choose B?
Because it's the palisade mesophyll cell,
Mm-hmm.
and they have a high
Amount
concentration of
of
chloroplast.
chlorophyll, beautiful. And that's where most photosynthesis occur, and they need enough carbon dioxide. The more carbon dioxide there are, the higher the rate of photosynthesis, and that's where most photosynthesis happen. Very good. What's the name of A, C, and D? What's the name of part label A, C, and D?
Um, A is epidermis.
Demo.
Then C is spongy mesophyll. Then
What did you say A is?
epidermis.
Epidermis cell, yes.
C is the spongy mesophyll, and
Are you sure C
D
is the sponge? No? C is this part of
oh, the vein.
yes, the vein, okay. Vein. Okay, I'll
Then D
accept
is
vein. Instead of vein,
D.
what are the veins there? The transport versus the xylem and what?
Uh,
Xylem and what?
xylem and no phloem. Hmm?
Phloem. Thank you. Yeah, so xylem and phloem. So any of these answers will work. And what's D?
And the stomata.
No, 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 no. This is still part of this. It's still part of the spongy world.
Spongy mesophyll cells.
It's still part of the spongy because you can see S spaces here. Okay, it's still part of the spongy mesophyll. All right. Let me add some more label here. That's E. Okay, that's E.
The lower
And F. So what's
epidermis.
E?
Lower epidermis.
Yes, what part of the lower epidermis is E?
Mm.
You can see a hole in between E and the other guy there, which I labeled as F. So what does that show you? Let me go back to let me go back to let me go back to what's it called? No, sorry, that's not it. Okay, so that's what I labeled E. So what's E showing you from that? This is what I labeled as E. And I said this space here is F.
The guard cell.
The guard cell. That space is what?
Stomata.
The stomach. Because it's one, is instead of stomata, you say it's what?
Stoma.
Because it's just one whole stomach. Alright, so take note of that. So E there is just one of the guard cells. This is the other guard cell. So there are two guard cells. The hole in between them becomes what? The stomach.
Oh.
Do you get now? So these details are very important. You have to really give eyes and ears to details. It's these details that will make you lose 1, 2 marks in between. And by the time you start losing 1 mark at one part, 2 marks here and there, you instead of you getting 100, you might end up getting 80 something. Not because you don't know, but you just didn't pay full attention to some of the details. And those details are what the examiners usually use to rob students of their marks. So please, everything being said is always very important. All right, that's why we make all these references to questions, to what's the word now? To reinforce the learning and to make sure you have the in-depth knowledge of what you need for the exam. Okay? Yeah, so that's that. Let me see if there's any other question here. I want something that has to do with those experiments in photosynthesis, and I've not seen anyone yet. Okay. Did you answer this one?
No.
Okay, let's see you try this.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have you know the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech at gmail.com, and they would reply to you, talk about your time zone, talk about and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable and you get the very best of service. We also have for the E levels as well, and also for the EdExcel. So we have for the Cambridge E levels and EdExcel E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at agile@gmail.com, and they'll reply to you. And the good news about this is what I've discussed with the sales team is we've tried as much as possible to see how we can subsidize the amount according to countries, so it doesn't really look very, very heavy. Okay, so this is really important. Yes, we do charge in pounds, but again, we try to be as much as possible considerate to your countries, and we don't just skyrocket the price. No, because at the end of the day, we know how it feels, and we really want you to actually get the best of service, and again, be able to afford it from the comfort of your home. So again, Ajiro Tech private tuition is currently ongoing. You can enroll yourself for the IGCSE and then for the E levels. Again, for the IGCSEs, these are the subjects, and for the E levels, these are also the subjects. So, again, what are you waiting for? Just send an email to the system. If you're an IGCSE student preparing for IGCSE, if you're watching this in 2025, watching this in 2026, watching this in 2027, we're currently ongoing, and this is actually a good time for you to actually explore and then get it tailored to your own needs. So trust me, you don't want to miss out.
So what's your answer? Honorable.
I'm deciding if it's A or B.
Mm-hmm. So what are you settling for?
Um, A.
A. Very good answer, A. Because since you are testing the leaf for starch, when you apply iodine on the leaf, okay, iodine in a part that does not contain chlorophyll, where there's no starch, will remain what? Brown yellow. So the white part is chlorophyll. So by the time you test it for iodine, it remains brown yellow. But the part that has chlorophyll, which is the green stripe part, will turn blue black. That's A is the most appropriate answer. B will be wrong because the white part, when you apply iodine, will not stay white. It will remain yellow brown or brown yellow. Okay, so you are very correct picking A. Yeah, that's what I wanted to be sure of, and you were done well picking the right answer. Okay, let's try question 8. It's still on the experiment now. Okay. Now the question is, what happens to the lime water in class X and Y when the pump is switched on? So air comes in this way. The pump pulls up the air as it enters this way. So sodium hydroxide, they told you, removes carbon dioxide from the air. And lime water usually goes cloudy if CO2 is bubbled through it.
Um, I'll, I'll go with C.
C. Are you sure you want to go with C? Yeah, give me the reason why you go reason for going with C.
Um, because for X, lime water gets cloudy if carbon dioxide has bubble through it, but then the
Mm-hmm.
sodium hydroxide removes carbon dioxide from the air, so it can't go cloudy if there's no carbon dioxide
Okay.
going
So
through
since
it.
sodium hydroxide removes carbon dioxide, it means this air passing through this way, we have no what? No what?
Oh, actually,
CO2.
let me go with D. Let me go with D.
D. Okay. All right. So we agree that for X it stays clear, right?
Yes.
Because since there's no CO2, it does not turn cloudy. Then the air goes all the way down to the plant in this. So in D it says this stays clear again. Why are you saying it stays clear for D?
Um,
For what?
because the tube that is inside the lime water in flax X is also going to the one that has the black pepper in it. So
Mm-hmm.
if the and also the one that is in flax Y is also going into the one that has the black pepper in it. So
Mm-hmm.
if the one that has the black pepper in it also has sodium hydroxide, it will transfer it to the one that is in flax Y.
Mm-hmm. No, look
So
at the
meaning
idea
that
here. This plant has a black what? What is used to cover the plant now? A black what?
oh, black pepper,
What does that tell you? No what
no photosynthesis.
is happening in this plant? One, there's no carbon dioxide, so no carbon dioxide is reaching the tube here where the plant is. And because this plant is the flax, the conical flax is covered with a black paper, light will not enter, meaning it can't carry out photosynthesis because one, there's no light, two, there's no CO2. So what will this plant do? What life process will it carry? It will only carry out respiration. The plant can only what?
Also.
Respire. Since there's oxygen in it. So the leaves, the cells will respire. And when they respire, what are the waste gas produced during respiration? What's the waste gas produced during respiration?
Um,
What waste gas is produced during respiration? Are you with me, go?
yeah, yes.
So what, since
Um,
this plant is only respiring, what is the waste gas produced during respiration?
moving um
Can you write out the equation for respiration for me, aerobic respiration? Or spell it out for me. Can you remember that?
uh um yes.
Okay, in aerobic
Um,
respiration, what
glucose glucose
mm-hmm.
plus oxygen um gives carbon dioxide plus water plus energy
Carbon dioxide, okay,
plus water plus energy ATP.
plus, okay. Energy is not part of the equation, okay? Energy is released during the process of the reactions happening. So usually energy is not part of the reaction as ATP. So you don't need to write plus ATP. It is the different
Yeah.
processes that release the energy. It's not part of the equation. Take note of that. Now, what I'm
Okay.
asking you now is, since this plant is carrying out respiration only, what waste gas is produced during this process in this plant? What waste gas is produced by this plant in this setup here? Since this plant is respiring, it means the leaves are releasing a lot of what? CO2 from respiration, because oxygen is passing from here to the plant, and the plant respires, and when they respire, they break down the oxygen to release carbon dioxide. It means carbon dioxide will pass from this plant all the way to this next tube containing lime water. And what does lime water do? It absorbs what? CO2. And when it absorbs carbon dioxide, what does it do? It goes what? Cloudy. So the carbon dioxide will be bubbled through this, and so it goes cloudy. So your initial answer is
Oh.
correct, which is C. In flask Y, it is clear, but in flask X, it is clear, but in flask Y, it goes cloudy. Because these plants can't carry out photosynthesis, since there are no carbon dioxide carried on for the plant, and there's no carbon dioxide in the air the plants get, they can but only respire. When they respire, they are producing CO2, which is bubbled through the lime water and makes the flax white to turn cloudy. So your answer is C. Do you understand that?
Yes.
Do you get the details now? So these are the intricacies involved in the questions that involve nutrition in plants. I think that's the most we can take at this time. That's the most we can take. Okay, let me try let's have you answer question 10 again. So I'll be sure you understand fully what you have explained. So let's go to question 10. It's having the same idea as the one we just answered. Yeah. Time up. It's a multiple choice question, so you have just about 45 minutes to respond to your answer. If you must answer all of your questions, so time is of essence. Speed and swiftness in deciding answers matters. Yes, Mirabel. I need to get your answer now.
Um, C.
Why are you going all the way to C? Mirabel? Why? Hey, that's way out of box. Give me the reason. Why did you go to C? Tell me why you are going to C. Let's go. I want to hear you.
Um, so, damn.
Go on, I can hear you.
So my reasoning is that C contains a water plant and is also exposed to light. So that
Mm-hmm.
allows photosynthesis to occur, which
Mm-hmm.
consumes CO2. So
Consumes
um
what? CO2. So uses up
the plants will use CO2 for
CO2.
photosynthesis of light.
Yes.
So that means that there will
Mm-hmm.
be a decrease in content.
Beautiful. 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 Oh, yeah, tell me why A is wrong, why B is wrong, and why D is wrong.
Um A contains has a water plant, a snail, but
Mm-hmm.
is covered in the black polythene, which prevents photosynthesis. So
Beautiful.
CO2 will increase
Thank you.
because of respiration.
Will
Then
increase
B,
because of
it will,
respiration. Beautiful. Both in the plant
yes,
and for the snail. Okay.
yes. So B, it has a water plant, but it's also covered. So it's the same with A, but removing the snail. Then
Mm-hmm.
D only has water and is exposed to light, so there's no plants to photosynthesize, so it will just remain the same.
Mm-hmm.
CO2 will remain the same.
I think it's going to remain the same. The snail will be respiring. And when snails respire, okay,
Oh, then
they
there's
will
no
produce some, yes. The snail will respire and they will produce carbon what? Dioxide. So the carbon
dioxide
dioxide will go here, it
increase.
goes up, here it also increases. So the only part where it decreases is C. So you are very correct. You are very correct. You are very correct. You are very correct. All right, I think you get the idea now, and that's what I wanted to be sure of. That's why I tricked you a bit. Then why did you go all the way? So with your reason, I'm sure you understand what it is. Okay, so
Mm.
that's good. That's good. I think that's just fine. I like your, at least I want to believe you have a full gist of what we have learned so far. So we'll be going to transport in plans. So we have just about 3 minutes or so to go. But let me just do an overview of what we'll be looking at now. So we'll be going to plant transport, okay? And in plant transport, we'll talk about the materials transported. In plant transport, we'll be looking at the materials transported in plants, okay? We'll talk about the vessels in plant. Okay, although we've mentioned some of it briefly as we were talking about photosynthesis, we'll look at the materials transported. We'll talk about the vessels or the medium for transport in plants. We'll look at the adaptations, okay, for their functions. Okay, we'll talk about where they get all these materials from, okay, and how they transport them, how season affects the materials they transport. We'll talk about the mechanism that ensures that these materials are carried from one point to another. Okay, the functions of all these transport vessels. So a lot of this we are going to discuss in our next class, and we'll also discuss some important terms that are associated with transport in plants. So thank you very much yet for your attention. I want to say you are doing very well, and hopefully as we go through the sections, you would be better off in your approaches to answering questions, and definitely you'll get better grades, which of course is what we want to see. And I'm sure you'll be happier when you see your grades improving. So, thank you. I think that's all we can take for now. And I see you next week.